So thank you everyone for joining Department of Medicine Grand Rounds this week. Dr. Weiss couldn't be here, but on behalf of the education folks at the Department of Medicine, thanks for joining for this important Barry Matterson Endowed Lectureship in Hypertension. Before our traditional chief resident introduction of our speaker, I wanted to give Dr. Preston a moment to tell us a little bit about this endowed lectureship in honor of Dr. Matterson. So Dr. Preston, please.
Sure, thank you. Dr. Barry Matterson was a widely recognized hypertension researcher and former chair of the VA Cooperative Study Group on hypertension. He was also a member of the JNC. He was also a wonderful mentor and colleague for many of us. He didn't want a big fanfare for these lectures, so he kind of instructed me in his own words to say that he established this endowed lectureship in perpetuity to bring leading hypertension experts to medical grand rounds. The purpose of this lecture series is to expose faculty, house staff, and medical students to prominent hypertension authorities and to provide the opportunity to posit relevant questions. And that's vintage Barry Matterson. And anyway, thank you very much, Dr. Rao.
Sure. I will be introducing our speaker today. So Dr. David Ellison is Professor of Medicine and Chemical Physiology and Biochemistry, Director of the Oregon Clinical and Translational Research Institute, and Associate Vice President for Clinical and Translational Research at Oregon Health and Science University. Prior to assuming these roles, he was head of the Division of Nephrology and Hypertension for 13 years. He's also a staff physician at the Portland VA Medical Center and the past chair of the subspecialty board in nephrology for the American Board of Internal Medicine and past chair of the American Heart Association's Council on the Kidney and Cardiovascular Disease. He was program chair for the American Society of Nephrology's Kidney Week in 2010 and was an elected member of its leadership council from 2018 to 2022. He is currently an elected member of the Association of American Physicians, the American Clinical and Climatological Society, and is past chair of the Kidney, Molecular Biology, and Genitourinary Organ Development Study section for NIH. He also was previously a standing member of the Renal Merit Review Study section for the Department of Veteran Affairs. Dr. Ellison is a leader in understanding how salt is reabsorbed by the kidney on the genetic basis of human hypertension and on diuretic treatment of edema. A long-term focus of his research is the thiazide-sensitive NaCl cotransporter. His early studies helped define the distal convoluted tubule of the kidney and how mutations of NCC can lead to Gittleman syndrome. Dr. Ellison has also led efforts that have established a previously unrecognized potassium switch in the kidney, which explains beneficial effects of dietary potassium intake and defines how mutations in a novel kinase pathway cause familial hyperkalemic hypertension. Finally, his group has demonstrated that the immunosuppressive drug tacrolimus causes hyperkalemia and hypertension by activating the NCC. This work has been translated to an ongoing clinical trial. All of his work melds basic and clinical approaches and has been published in top journals, including Nature Medicine, the Journal of Clinical Investigation, Cell Metabolism, and the Journal of the American Society of Nephrology. He is also a practicing nephrologist and a dedicated teacher and mentor to medical students, residents, nephrology fellows, and postdoctoral scientists. Welcome, Dr. Ellison.
Thanks very much. Let me see if I can share my screen correctly. Let's see. Share. And then. Are you seeing the slides or not?
No, we're seeing your background, but not the slides.
Okay, let me just fix this and I will. It was working before, but there. All right, now. Okay. Okay. Are you seeing my screen now?
Yes.
How about now?
Yep, perfect.
All right. Sorry about the I've been doing these virtual things for the last several years, and I still mess up, so I apologize for that. Well, thanks very much for the nice introduction, and I didn't know Dr. Masterson, but certainly knew of his outstanding work over the years in hypertension. And I think that the accomplishments of the Joint National Committee are really unparalleled in terms of how it's really led to the dramatic reduction in cardiovascular disease mortality in this country over the last 50 years. That's really been a great accomplishment in medicine. So I'm delighted to be here and talk about this. So I called my talk "Ending the Salt Wars: Diets for the 21st Century." And I'll tell why that is in a minute. I do have a couple of disclosures. Most of my work is funded by the NIH, the National Institutes of Diabetes, Digestive and Kidney Diseases, the Department of Veterans Affairs, and the Leduc Foundation. I was previously on an advisory board for a brief time for some preclinical development. None of this really has anything to do with what I'm going to talk about today. And importantly, I have never received funding from the food companies or the salt lobby. So anything I say today is my own opinion and not based on any associations. That doesn't mean I don't have opinions. So this is an outline of what I plan to talk about today. First, talk about dietary salt, potassium, and human health, giving a little bit of broad background, and then spend some time talking about the complex systems that regulate salt intake and salt balance. I am not a neurophysiologist, but I'm going to talk about some really amazing work that has really advanced how we understand how the body senses the need for salt and water and food. And then talk, as alluded to, about the relationship between potassium and sodium as we understood it back 11 years ago, and now as we understand it today. Talk about this potassium switch that we're interested in, and then end up by talking about dietary approaches to better health and why I think we may be on the precipice of really addressing some of the problems that have been around for a long time. So there's reasons for optimism in 2024. So many of you have probably seen this kind of data. I just chose some because it is representative and appeared in good journals. So on the top is a paper published in 2014 in the New England Journal of Medicine, which just looks at the relationship between dietary salt intake and systolic blood pressure. This is a large epidemiology study, and not surprisingly, back in 2014, everybody expected this. That's not why it was published in the New England Journal of Medicine, but there was a relationship between dietary salt intake and blood pressure, where people who were eating more salt in their diet had higher blood pressure, and people who were eating less salt in their diet had lower blood pressure. Not a surprise. That's epidemiology. At the bottom is a clinical trial. This is one of the many examples of the famous DASH diet trial. This is a follow-up study in hypertension of the original DASH studies, which were published also in the New England Journal of Medicine. So these are shorter-term trials, but these are where people were fed specific diets, and so they could actually monitor and prescribe dietary intake. And you can see that they had the same sort of relationship between dietary salt intake and blood pressure. This is mean arterial pressure, lower salt intake, lower blood pressure. So people, almost everybody probably in this room or virtual room would know that. What this led to over the last really many years is recommendations to reduce the amount of salt we take in our diet. So I just looked around to see how far back I could find recommendations, and this is just, I'm sure there were recommendations before this, but this is from 1977, and there was Dietary Goals for the United States, second edition, the Select Committee on Nutrition from the Health and Human Services. And so even back then, goal number 7 was to limit the intake of sodium by reducing salt intake to 5 grams a day. 5 grams of salt is about 85 milliequivalents of salt. And as I'll show later on, that's a lot less than we eat. So back when I was in high school, they were recommending that we reduce the amount of salt we eat. And so that's really been a consistent recommendation for many years. But I called this the Salt Wars because Other people have referred to it as the salt wars. This has been a very controversial recommendation. It's been around. The major august groups have recommended reducing salt intake, but then there was a article in the New York Times a number of years ago, 2012, saying, "Salt, we misjudged you," trying to argue that the data in support of reducing dietary salt intake was not as strong as most of the large groups had suggested, and really pushed back against it. And this led to a back and forth, some of it in the scientific literature, but a lot of it spilling over into the popular press. And so actually the term salt wars dates back to an article in Science magazine by this guy, Gary Taubes, who's a journalist. But then it was talked about by the head of the NIH. And so there was a book a number of years later by Michael Jacobson called Salt Wars: The Battle Over the Biggest Killer in the American Diet. And you can tell from the subtitle that Michael Jacobson came down strongly on the side that we all need to eat less salt, because that will protect us from cardiovascular disease and hypertension. So these are the salt wars. So, if the major groups have been recommending that we reduce the amount of dietary of salt we take in our diet, how are we doing in terms of doing that? Have we reduced the amount of salt in our diet? And the answer is no. And I want to see if I can get a laser pointer here. I don't know if you can see my laser pointer. Can you see it?
Yes.
Yes,
Good.
we can.
So this is just looking at dietary salt intake in kids, in men and women, and we can just look more on the right side. And this is through the years 1971 to 2003 and later. And you can see that since the time of the recommendation to reduce the amount of salt in our diet till now, if anything, the amount of salt we're eating has gone up, not down. So despite every recommendation to reduce the amount of salt we're eating, those recommendations are having no effect. And so I guess what I'm going to argue in the end is we need a new approach, because the past approaches have not been working, even if they haven't been working. So this is the current target for dietary salt intake in the country, which is about 2.5 grams of salt. And then a lot of recommendations include an ideal intake, which you can see is closer to that 85 milliequivalents recommended back in 1977, and that's like 1.5 grams of salt intake, and that's very, very low. So you can see that we're not close to any of those, and so we've really not made progress. So this is what I want to focus on a little bit, is in terms of, this is the same two studies, but adds another parameter to show the effects of dietary potassium to mitigate the effects of dietary salt, and why this may be a pathway to improved prevention and treatment of hypertension. So these are the same two studies. In the top is the epidemiology study, where when they compared the effects of different amounts of salt consumption versus different amounts of potassium in the diet. And they did this based on measuring the sodium and potassium in the urine as an estimate of dietary intake. And you can see that at every level of dietary salt intake, if you ate a high potassium diet, your blood pressure was lower. That's epidemiology. That's not a clinical trial. But you can see even more impressively in the DASH trial, when they did the DASH diet is a high potassium diet. And I should emphasize that the DASH diet has other differences from the control diet. It's not just high potassium. But many people believe, and I think there's increasing evidence for the fact, that much of the beneficial effect of the DASH diet to lower blood pressure is the high potassium. So this is a high potassium diet, and you can see two things. One, it lowers blood pressure at every level of dietary salt intake, but it's most effective at high dietary salt intakes. In fact, we often talk about salt-sensitive hypertension as if it were a thing. But in fact, salt sensitivity is not fixed. It can be altered by other dietary constituents. So if you're on a high salt diet, increasing potassium intake lowers blood pressure a lot. If you're on a low salt diet, it doesn't lower it that much. So its efficacy really depends on the amount of salt you're eating. It's clearly an interaction effect between the two things. So how does this happen? So this is the thing that many of you may have seen, which was published in the New England Journal of Medicine just, just a couple years ago, which was got a lot of attention, which was effective salt substitution on cardiovascular events and death. This is a study led by Bruce Neal and collaborators, but it was conducted in China he had a bunch of Chinese collaborators. And what they did is they had almost 21,000 participants from 600 Chinese villages, and 300 of the villages were provided regular salt for cooking, and 300 of the villages were provided salt substitute, which contained 75% sodium chloride and 25% potassium chloride. And they got an amazing follow-up of 99% for 5 years. And you can see down below that this is the mean difference in blood pressure. So there was a lower blood pressure in the group that was randomized in the villages that were randomized to the salt substitute by 3 or 4 millimeters of mercury. So that kind of wasn't that surprising, and that was what they were hoping they would find, which is the effect of replacing 25% of dietary salt with potassium, lower blood pressure. So probably wouldn't have gotten in the New England Journal of Medicine just for that. Oop. But here's the more impressive result, which is that there was a statistically significant reduction in the primary endpoint, which was stroke. So these people, the people who were selected for the study, were not just everybody. It was people at somewhat high risk for cardiovascular disease. So somewhat older individuals with a history of some cardiovascular disease. And you can see in that population, there was a reduction in the risk of stroke over 60 months. So that's not that long a period of time. So the relative risk was 0.86, which had a p-value of 0.006. And then a secondary endpoint was all-cause death. And you could see that the rate of all-cause death was also reduced by about the same amount, which is the first time there's been a demonstrated benefit in terms of all-cause mortality of doing a dietary intervention like this. So this really suggested, but I'll come back to some caveats about this later on, that replacing regular salt with a salt substitute that contains potassium might be beneficial in heart outcomes. So is it the salt reduction or is it the potassium increase that led to this effect? Well, there's a number of follow-on studies or papers that provided evidence that really was potassium, but I think you can see that the salt intake was reduced by only 8% by this intervention, but potassium intake was increased by 55%. So just looking at the percentage changes in diet, it seems more likely that it was the higher potassium intake that was having the beneficial effect. So salt handling is complex. This is from a paper we published a couple of years ago in the New England Journal of Medicine. I'm not going to go through most of these systems. I just want to point out that there's all sorts of systems involved in sodium homeostasis and potassium homeostasis. It involves the nervous system. It involves other systems as well. I'm not going to spend a lot of time on those, but it is a very complex regulatory system. But I want to talk for a minute about salt appetite. And this is not stuff that we've done in my lab, but there's just been some really outstanding work that really has elucidated why we eat salt and what drives us to eat salt. So I don't know if any of you guys follow Joel Topf, whose handle is Kidney Boy. But he's a very well-known educator in nephrology, and he posted this a couple of years ago. So this guy said, the most private thing I'm willing to admit is I'd like to drink pickle juice. And so Joel Topf said, this post on BuzzFeed makes me so glad I never suffered the indignity of online dating. My favorite is this guy. So he says, what are the odds he has Barter's or Gittleman syndrome? Every patient I've encountered with Barter's or Gittleman syndrome has admitted to drinking pickle juice. So it's sensitive, but I don't know how common it is. Turns out it's very common. So for people with salt-losing diseases like Gittleman syndrome, their brains tell them that they're volume short. And so that when they get home, they want to go to the refrigerator and drink pickle juice because that's a high salt food, and that restores their extracellular fluid volume. So how does that work? as I mentioned, there's been a absolutely brilliant series of papers published in the highest impact journals, Nature and Cell. They've really elucidated the physiological basis for salt and water appetite. And what these several groups have done is taken very simple animal models and then used very sophisticated techniques to unravel the causes. So their simple model is as follows. They'd have mice, and they have a control mouse. They have a mouse that has an osmotic stress, and they do that by injecting hypertonic saline into the peritoneum. Then they have a mouse that's volume depleted, and they volume deplete those mice by giving them furosemide. So that happens in our patients as well. And then they, excuse me, have mice that have pure water depletion, and that they just don't feed them any water for a couple days, and that's how they get water depleted. So it turns out that, and maybe you think this is obvious, but when you do this to mice, and then you offer them either saline to drink, like the pickle juice, or you offer them water to drink. So you have both bottles, and the mice can choose what to drink. If you do an osmotic stress, so you make them hypertonic by infusing hypertonic saline, then all they drink is water. So the gray bars is drinking water. The red bars is drinking saline. So they did it with either hypertonic saline or mannitol. So they drink water. Well, what if you make them volume depleted? So then they're depleted of both water and salt. And those mice will drink both salt water and pure water because they're depleted of both. And then if they're water deprived, they get mostly water depleted, and therefore, again, they'll drink water rather than salt solution. So this is actually, I think, right up the road from where you guys are. That was invented Gatorade for the Florida Gators, and that was invented by a renal physiologist, and they were tapping into this. They knew that when you exercise, you develop a salt thirst because you're getting depleted of salt. And this is why I think Gatorade actually, to me, tastes good if I've been sweating a lot, but if I'm sitting around, it tastes terrible. And that's because I don't want to drink a salty fluid. So the mice drank the right thing to replace the deficit. They have some physiologic system that allowed them to do that. So then they did this sophisticated analysis of their neurons in the brain. And without going into detailed results of those studies, they found that if you were thirsty, you had some neurons activated. If you had a salt depletion, you had other neurons activated. And actually, if you were hungry, you had different neurons activated. So then what they were able to do is they were able to use optogenetics. And so they expressed optogenetic molecules in the brain only in one set of neurons. And so then they could activate those neurons by turning a light onto the brain. So you can see what happened. So these mice are just sitting around, but they have these optogenetically expressed genes in the brain. And if they put them in the water neurons, and then they turned on the light, you could see that the mice started drinking water right away. They didn't drink any salt water, any sodium. On the other hand, if they put the genes in the salt neurons, they started drinking salt right away. And then if they did it in the food neurons, that actually takes longer to get stimulated, but then they start eating. So we have in our brain neurons that tell us I'm thirsty for water, I want salt, or I want food. It's not, in some ways, not surprising, but it's pretty elegant. Then they went on to talk about other aspects of these physiological systems. So, if you're hungry or you're thirsty, you want to satiate those needs. And they studied how this happens. And so it turns out that when you turn off the light and turn off these neurons, for the water neurons, you stop drinking water right away. But it turns out there's actually a gulping reflex. So it actually turns off before you've actually changed your body constituents, because when you gulp water, the brain senses that you're satiating your thirst. The same thing happens with salt. When you take in salt, the salt neurons get turned on right away. It turns out, like everything else, that the food neurons are a little bit slower, but they get turned off eventually as well. So this is one of the reasons that we're kind of driven to eat salt. And it's hard for us to reduce the dietary intake of salt, even though everybody is telling us to do it. And because all the food companies are putting a bunch of salt in food because they know that that's going to make people eat it more. So where does the salt go? This is an area where it's also been some recent progress, more fully understood than it was before. So this is total body salt content, and you probably learned this in medical school, but most of the salt in the body is in the extracellular fluid, and most of that, you may not have learned, that is in the interstitial fluid here. Yep, there it goes. Interstitial fluid here. There is some salt in bone, but this is what we call non-exchangeable. So this is sort of fixed in the bone and not accessible to being added or taken away. But the other thing we've learned recently is the salt in the interstitial fluid is, some of it is bound to these glycosaminoglycans, and so that's a sort of special compartment of salt in the interstitium, which may be important for blood pressure control. And this is from a diagram we put in that New England Journal review a couple of years ago that just shows we've learned a lot more about how the salt and fluid, this is where edema forms if you accumulate too much salt in your body. And it turns out that we've learned a couple things recently that have changed how we think about this. The first is that when I was in medical school, I was taught that because of the Starling forces, when blood comes into the capillaries, there's outward movement of fluid, there's filtration of fluid at the inset of the capillaries, but then fluid comes back in because of the higher oncotic pressure in the venous side. It turns out that's probably not true, at least under most circumstances. And we now believe that there's actually fluid transudation going on throughout the capillaries, and that all of that fluid that accumulates needs to be returned to the body through the lymphatic vessels. And then the other thing we've learned is that there are these glycosaminoglycans, which bind up a lot of the salt here in the interstitium and can act as a depot. And the reason that the interstitium is so important is because, unlike muscle down here, where you have closely packed muscle fibers, there's not much room in the for interstitial fluid. Here in the skin, you have all this free fluid spaces. So we accumulate a lot of fluid in the skin, and this is why, when you have patients with edema, it's mostly under the skin is where the edema is. All right, now I want to turn to potassium and move along and talk about potassium and how it impacts sodium balance. So back in 2003, this was a paper from 2003, not that long ago. This is what we understood about how potassium was handled by the kidney. This may have been what you learned in medical school. So potassium is filtered. We filter about 600 millimoles per day. Potassium is reabsorbed along the nephron, and we reabsorb almost all of the potassium before you get into the distal tubule. And in the distal tubule and collecting duct, potassium is secreted. So most of the 90 millimoles of potassium that is excreted comes from potassium secretion here in the connecting tubule and collecting duct. And back in 2003, we actually knew something about the molecular basis of these processes. So here in the connecting tubule and collecting duct, we have principal cells, which are the main cells that are responsible for this. And you may remember from medical school that these have the epithelial sodium channel, or ENaC, at the apical membrane, and it also has a ROM-K potassium channel, and that's by, in the distal tubule, when you reabsorb sodium, you secrete potassium. So this was all well known back in 2003. So what's happened over the last 25 years, one of the things that's happened is the molecular basis of a number of rare monogenic diseases has been solved. Almost all of these monogenic diseases of the kidney are now solved, and we know what the causes are. So I started out by showing the guy who might have Gittleman syndrome, and then there's another syndrome called Gordon syndrome, or familial hyperkalemic hypertension. And these are kind of mirror images of each other. So in Gittleman syndrome, it almost always presents with hypokalemia, low blood potassium. It's an autosomal recessive disease. It has metabolic alkalosis. And importantly, the blood pressure tends to be low. Renin levels are high, hypocalciuria is common, and hypomagnesemia is common. And it kind of looks like what happens when you give high doses of thiazide diuretics. Gordon syndrome is kind of the mirror image because hyperkalemia is always present. It's autosomal dominant. You have metabolic acidosis rather than metabolic alkalosis. Blood pressure is high rather than low. Renin levels are low, and there's hypercalciuria, and you can treat it by giving thiazide diuretics. So we now know what the molecular. I'm sorry. We now know what the molecular basis of both Gitelman syndrome and Gordon syndrome is, and they both affect the distal convoluted tubule cell. So Gitelman syndrome is caused by a loss of the sodium chloride cotransporter, which mediates thiazide-sensitive sodium chloride uptake into distal convoluted tubule cells. It's actually mutations in the gene for NCC that cause the disease. The opposite is Gordon syndrome, which isn't caused by mutations in the thiazide-sensitive sodium chloride cotransporter, which I show here, but is caused by mutations in genes that activate the sodium chloride cotransporter. So the mutations that cause it are these Wnt kinases that activate the transporter here in the apical membrane. So you have a salt-losing disorder, Gitelman syndrome, and a salt-gaining disorder, Gordon syndrome, and these are mirror images of each other. So this cell transports sodium and chloride and not potassium, but I started out by telling you that the disease presents with disorders of potassium balance, and the disorders of sodium balance are a little bit secondary. So what does this have to do with potassium balance, and why does it cause changes in potassium balance if it only affects the sodium chloride cotransporter? And these were a series of studies done a number of years ago. This is the first done by Volker Vallone from UC San Diego. And he showed, importantly, a couple of things. One, we had actually shown before this that low sodium diet stimulates the thiazide-sensitive cotransporter. So this is NCC activity. This is diet. Low salt diet stimulates NCC. That was known. But what Volker showed is that a low potassium diet also stimulates NCC. So this is high potassium intake, this is low potassium intake, and you can see that NCC is stimulated by low potassium intake. And so a Western diet, or what we call a Western diet, or the diet you probably eat in Miami, is a high salt diet and a low potassium diet. And so these should have opposite effects on NCC. The low potassium should stimulate NCC, the high sodium should inhibit NCC. So I had a graduate student a couple of years ago who asked the question, which wins in this situation? Does the potassium have more potency in controlling NCC, or does sodium have more potency in controlling NCC? So he designed some very nice experiments in mice where he put mice on a high salt diet and then either normal K or a high salt diet and low K. And again, this is the sort of Western diet. And you can see when he measured NCC activity, there was a huge increase in NCC activity in mice that were consuming a Western diet or a high salt, low potassium diet, suggesting that potassium beats sodium with respect to NCC. So this is how we now understand potassium transport in the distal tubule. And again, I'm going to come back to the fact that this cell doesn't transport potassium. We understand that low plasma potassium actually stimulates these cells through this Wnt kinase pathway, and you don't need to remember the details of this, to turn on NCC. And this looks like the most potent way that you can turn on your thiazide-sensitive sodium cotransporters. So a high potassium diet actually acts like a thiazide diuretic. But this cell still doesn't transport potassium. So what does this have to do with potassium balance? And here we have to talk about the so-called aldosterone paradox and talk about the adrenal gland for a minute. So I think you all know what the adrenal glands do, but just to remind you of a couple things, one of the things the adrenal glands do is they make aldosterone, which is an important hormone for regulating salt-potassium balance. And one of the things that stimulates the production of aldosterone is angiotensin II, which is elaborated when you have dietary salt deprivation or your volume short. Whoops. But I mentioned the aldosterone paradox, which was is a phenomenon that was postulated by Mitch Halpern a number of years ago, where he asked the question, how does aldosterone, a single hormone that binds to a single nuclear receptor, serve two different physiologic functions? Kaliuresis when plasma potassium is high, and antinatriuresis when extracellular fluid volume is low. And I think part of the answer to that comes from another stimulator for aldosterone secretion, namely high plasma potassium. So let's look some more at that. So turns out this is from very old paper from Gordon Williams in the 1970s, showing that this is serum potassium and this is plasma aldosterone. And you can see this is people when they're eating a 10 milliequivalent sodium diet or a 200 milliequivalent sodium diet. The effects of potassium are very evident on both diets. So here's the effect of plasma potassium when you're eating a low salt diet. And here's the effect of plasma potassium when you're eating a high salt diet. Potassium clearly has an important impact on aldosterone secretion. So here's what we think of as the potassium switch now. And this is how we think it works. You have in your distal nephron a distal convoluted tubule and a collecting duct and a connecting tubule and collecting duct. I showed you a picture recently of the DCT, and I showed you a picture in that figure from 2003 of the collecting duct. And these two cell types work together to modulate sodium and potassium balance. So if you're on a high potassium diet, so if you have a high-ish plasma potassium, that tells the adrenal gland to secrete aldosterone, which goes to your collecting duct and it turns on ENaC. At the same time, the high-ish plasma potassium goes to the distal convoluted tubule and turns off NCC. Remember, I said that high potassium, eating a high potassium diet is like taking thiazides. So you've turned off NCC and turned on ENaC. What does this do? This means that you don't absorb sodium with chloride here. You deliver the sodium more distally, and ENaC is turned on, and so you absorb the sodium here in the collecting duct in exchange for potassium. Now, because you've turned off NCC, you don't reabsorb as much sodium in the kidney as you do at baseline. So a high potassium intake causes kaliuresis, because whenever you reabsorb a sodium here, you secrete a potassium here, but it also causes a natriuresis because you've turned off NCC. So that happens on a high potassium diet. What happens on a low potassium diet? Well, what happens on a low potassium diet is kind of the opposite. So in a low potassium diet, you have a lowish plasma potassium that goes to the adrenal gland and turns off aldosterone production. And when you don't have any aldosterone around, ENaC is turned off here in the collecting duct. At the same time, a lowish plasma potassium stimulates NCC and turns this on. So now all this sodium that's coming down into the lumen gets absorbed together with chloride rather than in exchange for potassium. So a low K intake causes potassium retention because you reabsorb the sodium here and not here, but also causes more sodium retention. And we think that's why it raises blood pressure. And so what about on a low salt diet? What happens then? On a low salt diet, there you turn on angiotensin 2, that goes to the adrenal gland and tells it to secrete aldosterone, and it secretes aldosterone. But through a different mechanism, angiotensin 2 turns on NCC. So in this case, you've got NCC turned on and ENaC turned on, and you just basically reabsorb all of the sodium you can. So a low salt intake causes salt retention, but this is physiologic, homeostatic, without a change in salt balance. So this is what we call the potassium switch. So this is mice studies. Does this happen in humans? Well, we think it does. And this is from some of the human data that I'm going to show you that. So this is a nice meta-analysis of randomized controlled trials where different groups have added potassium to the diet and seen what happened to blood pressure. And so this is added potassium in the diet, whether they've added 0, 30, 60, 90, or 120. And you can see there's sort of this U-shaped curve where if you add, let's say, 45 milliequivalents of potassium or millimoles of potassium to the diet, you've reduced blood pressure, but there's sort of a U-shaped curve. So if you eat a whole ton of potassium, you actually increase blood pressure. And that's true of both diastolic and systolic. But it clearly looks like adding some potassium to the diet is beneficial. And this is from the same paper showing that this actually interacts. This comes back to what I showed you about the New England Journal paper of the at the beginning, that this partly is if the effects of potassium are most impressive if you're on a high salt diet. So here what they've done is they've compared the effects of potassium in the diet to those people who are having a high salt diet, a medium salt diet, or a low salt diet. And this is grams of sodium, not salt. So on a low salt diet, you have this U-shaped curve. So there, if you add potassium, if you add more than about 60 millimoles, it ends up raising blood pressure. But here, if you're on a high salt diet, no matter how much potassium you add, blood pressure is low. And we think this is because aldosterone competes with the effects of potassium on NCC, and it's sort of the balance between the two. And so there's got to be a sweet spot here where you have the best ratio between sodium and potassium, and therefore get the most bang for your buck, if you will. So one other point I want to make is we think that potassium deficiency in potassium intake may actually explain some of the disparities in blood pressure that are well recognized. So it's been known for a long time that that non-Hispanic Black individuals on average consume less potassium than non-Hispanic whites or Hispanic whites. And so you can see this is non-Hispanic Blacks, and this is non-Hispanic whites, and this is potassium consumption, all males, females. So you can see, and this has been seen many times. So lower potassium consumption amongst certain demographic subgroups. And so Kurtz and collaborators a couple of years ago published a nice study where they looked at the effects of dietary potassium intake on the salt sensitivity of blood pressure, and they looked at the difference between Blacks and whites, and they found out that in those studies where people increased potassium intake, the difference in salt sensitivity between racial groups really almost disappeared when you had them on equal potassium intakes, suggesting that some of these racial and ethnic differences in blood pressure may be really owing to differences in dietary intake. And this is an area of active investigation. So getting close to the end now, but you may have seen, some of you may have seen, that last year, the FDA had a proposed rule to permit salt substitutes to be used in processed foods as part of the processed foods in an attempt to address the goals of reducing salt intake, but also perhaps increasing potassium intake. And you might think this was a no-brainer based on what I showed you before, but it turns out that some patient advocates, and especially people from the kidney community, were concerned because when I'm wearing my nephrology hat, we always tell our chronic kidney disease patients to stay away from potassium because there is a risk of hyperkalemia. And so certain patient advocates have warned the FDA that the proposal could do fatal harm to people. So actually, this is going to be discussed extensively over the next year. And in fact, the ASN is probably going to convene a group to discuss the pros and the cons of the FDA's proposed rule to permit adding salt substitutes to processed food. But this is sort of what I think where we maybe should be moving, but also some caveats to taking this at face value. One thing that I don't think so what I've shown here is sodium intake here on the abscissa and potassium intake on the ordinate. And a couple things I want to point out. The DASH study investigators kind of did something a little tricky that they don't talk about that much, which is their DASH control diet was actually made to be a low potassium diet. So it's the 25th percentile of dietary potassium consumption that they use for the DASH control diet. So when they say going to the DASH diet, changes blood pressure a lot, it does, but partly because it really increases potassium. So this is the DASH high salt diet, the DASH medium salt diet, and the DASH low salt diet. They're all very high in potassium, but the DASH high salt diet is pretty close to what this is the current intake in the US. So we're eating a lot of salt, but we're also eating more potassium than in the DASH diet. And so, so this is what the DASH diet is recommending. This is where we are right now. What about the New England Journal study that showed the impressive effects on mortality? What turns out in those villages in China, they're eating a heck of a lot of sodium and very, very low potassium intake. So you might expect the increasing potassium intake to have a very large effect. I didn't show it, but there was another study published in Nature Medicine that showed very similar results from Peru. And also those people consumed a very low potassium intake. So this is where we are in the United States. But I would suggest that if we, instead of making crazy recommendations that nobody can adhere to, if by changing to potassium containing substitutes, we could reduce the salt intake by 20 and increase potassium intake by 20 and get to here, we wouldn't be in nirvana, but we might be a lot closer to having an effective approach to reducing blood pressure and improving cardiovascular morbidity and mortality. Whoops. There we go. So in summary, what I've talked to you about is substituting some sodium chloride with some potassium seems like it could be a win-win. I didn't show you, but actually salt substitutes taste, pretty not bad. I actually have used them myself, and I think they're pretty much okay. Salt substitutes with potassium are effective, and they seem quite palatable, providing an opportunity for the food industry to do good and to do well at the same time. And, they don't want to put potassium substitutes in their food if nobody eats it. And so they need to make sure they keep getting the people buying the food. But I think that that's a possibility. So in the US, this will likely require some changes in food processing. The FDA has proposed this, but there are doubters, and I think we have to work that through. The potassium switch cassette comprises the potassium regulated thiazide sensitive sodium chloride cotransporter and the aldosterone regulated ENaC, which work together to jointly achieve potassium homeostasis, but also to affect blood pressure. A balance between optimal sodium and potassium intake keeps aldosterone in check, and we should continue to advocate for making fresh and less processed foods widely available. Even if we change the constituents in processed food, we should continue to advocate for a healthy diet in other ways, because in reality, the DASH diet was much more than just a high potassium diet. It was an overall healthy diet, and we should encourage that as well. So with that, I will thank you for your attention, and I would be happy to answer any questions.
Thank you so much, Dr. Ellison. That was fantastic and brought together a lot of stuff that we thought we knew from medical school, which was really helpful in some of the newer concepts in sodium potassium. I'll start by asking Dr. Preston if he'd like to ask anything.
Sure. Thank you for fantastic grand rounds and also for all the incredible work you've done over the last years in the distal nephron and the thiazide sensitive cotransporter. In addition to the very compelling evidence for this thiazide-like effect of potassium on the sodium chloride cotransporter and potassium-induced natriuresis, there are a couple of other mechanisms for the lowering of blood pressure that are out there that are still talked about. One being a direct effect of potassium on endothelial cells, and the other being perhaps a reduction in sympathetic nerve activity, specifically to the sympathetic intervention of the kidney, as also mechanisms. What do you think about the roles for these other two mechanisms?
Yeah, great questions. And, for the sake of time, And partly because we haven't done as much in those other areas, I didn't go over those, but clearly it can be this and that. And I think that the idea of endothelial mediated effects may play a role. These changes in dietary potassium doesn't change your plasma potassium very much. So we need to figure out how that's working. The sympathetic thing I think is really interesting, because the other thing I didn't have time to go through, but clearly higher potassium intake inhibits salt reabsorption in the proximal tubule as well. And as we now know from the SGLT2 inhibitors, which seem to cure everything, part of the mechanism of them is by inhibiting sodium reabsorption in the proximal tubule, you activate the TG feedbacks, the tubuloglomerular feedback system, which decreases GFR, but it also turns off the sympathetic nervous system. So one of the mechanisms by which potassium may be turning off the sympathetic nervous system, which is clearly good, is by activating the TG feedback system because of proximal effects as well. Again, there could be direct effects on the sympathetic nervous system as well. And in the chat, someone's asking, do you think the effects of the sodium potassium diet on stroke mortality is mediated totally through blood pressure lowering? And there's old data, and I'm blocking on who it's from the 1950s and 60s. I guess it's from Dahl, perhaps, suggesting that high potassium intake independent of blood pressure is beneficial to stroke, and did a lot of studies back then that suggested that. Now, I would say I'm a big believer that in science we make type 2 errors frequently, and it's very easy to miss a 1 or 2 millimeter reduction in blood pressure and declare blood pressure reductions to be not significant and therefore not important. So I think we have to keep in mind that we may miss it, but I think there is a lot of evidence that there are other effects, as you mentioned, in other organ systems that suggest that a high potassium intake is beneficial.
But Dr. Ellison,
Thank
just
you.
to combine a couple of the questions that are on the chat,
Yep.
I think one of the challenges you showed some beautiful tables and graphs about sodium potassium intake and milliequivalents, and I think when you're in the clinic trying to translate that into what that means into what specifically in my diet should I eat more or less of. Dr. Unmute if you'd like. He actually has had some roles in the FDA as well over the past many years in the GI space. He asked, for example, tongue in cheek, if an apple a day can be substituted for an orange or a banana a day. But maybe I can take that question. Please feel free to answer that. But also more broadly, how do you translate some of these concepts into the clinic when people say, well, what does that mean in terms of my salt to potassium intake? And do you have sample diets for them to follow?
I think it's really hard because, that what I talked about in terms of drive, people like to eat salt and it makes food taste good. So the one potential benefit of the salt substitutes having potassium is that they really taste pretty good. And so in theory, you can eat things and it'll be better for you. Some people have complained we should be doing, giving everybody fresh fruits and vegetables and not doing this stuff. But I can tell you, living in Portland, which was recently rated the best restaurant city in the country, that all of those restaurants use high amounts of salt. It just, you're not going to get a good review in the New York Times by having a low salt restaurant. So I think thinking of ways that we can do well and good are appropriate. Now, let me just get to the apple day versus a apple or apple plus, an orange and banana. You could talk about, I think nutritional epidemiology is a very challenged field, but one thing I believe is that the increase in sugar intake has been bad for society. And there is no question that oranges and bananas, while having potassium, have a lot of sugar. And how you balance that out is really a challenge. So I don't know what the right answer is, but I think maybe if we get people, if we start going in the right direction, recommend good diet, but know that most people, especially people who have challenged circumstances, who are working night shifts and stuff, it's really hard to eat fresh stuff all the time. And so I think working in multiple fronts at the same time, I guess that's my bottom line. So sort of not a very coherent answer, but I'm trying.
No, that's great. Thank you. And then Dr. Martinez had asked, we use, for example, urine studies, sodium, potassium for people in salt restriction, ascites, and other diseases. Any role that you can see in terms of dietary guidance of monitoring urine potassium sodium ratios?
We do that as a sign. I mean, in the laboratory, we do that as a sign for activation of the aldosterone system, and in our laboratory stuff, it works really well in terms of monitoring intake. I think you could make a case for that. We do it for we've done it for if you have a patient with hypertension and on a thiazide diuretic, and you want to know if they're, or with heart failure and on a loop diuretic, and you want to know if they're eating too much salt, we often recommend measuring a urine sodium. I think that makes some sense. And you could probably do it with a spot urine sodium ratio, because doing it 24 hours is a drag. So, I'm sure there's somebody's written a paper on that. It makes some sense to me. I haven't personally practiced that, but I think that's a good idea.
Well, on behalf of the whole Department of Medicine, thank you so much for a fantastic grand rounds in honor of the endowed lectureship from Dr. Matterson.